everyone. This is our first week talking about proteins, and we'll have 3 lectures on proteins. So today we're going to talk about classification and functions, measurement of protein quality, and digestion and absorption. So our first part of the lecture is on classification of proteins and their functions. So the objectives are to list the different ways to classify proteins, list the different functions of proteins, understand deamination and transamination, and define carbon skeleton and alpha keto acid, including their functions. Okay, so protein intake in the United States. The dietary guidelines recommend, for Americans, 2010, recommend a certain range of calories that we get from protein. So this includes a smaller proportion of calories from protein for younger children who really need more fat. And then for older children and adults, the recommended intake is 10 to 35% of calories coming from protein. Now, another way that we can calculate protein needs is by kilogram of body weight. And in the United States, we recommend 0.8 grams of protein per kilogram of body weight per day. So let's just do a quick little exercise. Let's calculate the needs according to these two methods for a woman who's 150 pounds and eats 2,000 calories a day. Now we see that if we calculate it for according to her body weight, her recommended intake is about 55 grams per day. If we use the AMDR, her recommended intake is 50 to 175 grams of protein per day. So you can see when we do it by body weight, we really get a recommendation on the lower end of the AMDR. So in actuality, in the United States, both men and women consume about 15.5% of their calorie intake from protein. So recommendations are to consume a wide variety of foods, pretty standard in the dietary guidelines. So we want high quality animal, dairy, and plant-based proteins. We see in the United States we have a much higher proportion of protein that we get from animal foods and dairy compared to plant proteins. So in the US we get 30% of our proteins from plants, while worldwide the average is about 65% of proteins come from plants rather than animals. And our intake has remained relatively consistent over the years, and as we all know, protein has 4 calories per gram. So we do carry quite a bit of our weight as protein in our bodies, with our biggest stores in our skeletal muscle, followed by protein that we find in our skin, in our blood, and then our liver and kidney. Though proteins are ubiquitous throughout our entire bodies, and we find them everywhere in the body. So the distribution of proteins among organs does vary with developmental age, with newborns having much less muscle mass, so less proportion of their protein is in their muscle. Okay, well, proteins are a very, very broad macronutrient. They have a lot of different functions and a lot of different properties, and therefore we classify them in different ways. So we can classify them by their structure, their solubility, their essentiality, nutritive value, and metabolism. So the first way we classify them is by their structure. So you can see we've got the structure of just a standard protein here, where we have our carboxyl group, our amino group, and a side chain here. And it's the structure of the side chain that depends how we classify the structure of the protein. So for aliphatic side chains, such as branched chain amino acids, we can see that the side chains only have carbons and hydrogens, and just like the carbons and hydrogens in fats, this makes these proteins hydrophobic. So it's really the properties of the side chain that really show, or how should I say, control what the functions of the protein are going to be in total. So the side chain can have a hydroxyl group, a sulfur group, they can be acidic or basic, or have aromatic rings. So proteins can also be classified by their solubility, though it's not as kind of straightforward as when we think of solubility of carbohydrates. So the main groups that we group them in for solubility has to do not really with how soluble they are in water, but really if they're simple or a conjugated protein. So simple proteins are made of amino acid units only that are joined by peptide bonds. So they're just amino acids. And we have some examples here. So we have the albumin and globulin that we find in egg whites and in our blood. We have prolamine, which is found in corn. Histones, which are found in our nucleoli and important for DNA synthesis. We have protamines and albunoids. And these are just some examples of simple proteins, which are molecules made up of amino acids only. Okay, and then we also have our conjugated proteins, where the protein is joined to another type of molecule or macronutrient. So first we have our glycoproteins and mucoproteins. So the glycoproteins occur where sugar molecules are covalently bound to proteins, and we've talked about this a lot before, that these glycoproteins are really important for, especially for cell signaling. And there's different types of covalent linkages that we see between the sugar and the amino acid, but the most common is an N-glycosidic linkage between asparagine and the sugar. So, okay. And of the 20 amino acids, only 4 that are listed here really bind to carbohydrates. So those are the proteins that we're going to find in our plasma membranes that are binding to the carbohydrates for these functions. Another type of conjugated protein is our lipoproteins, which we talked a lot about in the fat section. So of course, these are our LDL, our VLDL, our chylomicrons, and our apolipoproteins. And then our third type of conjugated protein, or sorry, there's more than that. Then we have our nucleoproteins, which are nucleic acids joined with proteins. And these are the main constituent of, or component of, viruses. Okay, next we have our phosphoproteins, where the protein is joined with a phosphate group, and this is present in milk casein. We have our hemoproteins, such as hemoglobin, where iron is bound to the protein, and that's shown in this picture here, where we have our 4 peptide chains and our heme groups that are bound within the protein chains. We have our metalloproteins, which is a protein that's joined with a metal, such as copper or zinc. And we have our flavoproteins, which have the FAD molecule joined with protein. Okay, so next is classification of proteins by essentiality, and this is one I think we're a lot more familiar with. So we classify proteins in this way by whether they are indispensable or dispensable. So an essential amino acid is an amino acid that we can't make in our bodies from other proteins, and so we must get them through our diets. Whereas a non-essential amino acid we can produce in our bodies from other substrates. So in addition to a protein either being essential or non-essential, it can also be considered conditionally or acquired essential. So this would be a protein that's usually a non-essential protein, but there's some physiological condition that is making the protein essential. So this could happen, for instance, if an organ fails to function properly. And we see that glutamine becomes an essential amino acid in advanced liver disease because we can't produce the glutamine if our liver is not functioning properly. And then a cirrhosis impairs phenylalanine and methionine metabolism. In severe stress, we can't produce enough glutamine to meet our needs, so we need to get an extra from the diet. And then there's a few other examples that you can read through here. Another classification that we're probably all familiar with is classification by nutritive value. So does the food, the protein food, contain all of the essential amino acids that we need? Excuse me, this should be essential amino acids. So does the food contain all of the essential amino acids that we need, or is it missing some essential amino acids? So a complete protein would contain all of the essential amino acids, and these are shown here. And these are foods that are complete proteins are typically of animal origin. So we have our eggs, cheese, meat, milk, fish, and poultry. Incomplete proteins are foods that do have some essential amino acids, but don't have all of them. And these are foods that are primarily of plant origin, such as legumes and vegetables and grains. Now, food that has incomplete proteins can be joined so that they provide a complete protein. So you really want to, if you're for instance, if you're a vegetarian or a vegan and you're not getting these complete proteins, you can address that by combining certain foods so that they're complementary and that together they provide all of the essential amino acids. So here's a table that demonstrates how we can pair foods together so that we get all of our essential amino acids and the food becomes a complete protein food. So if we have legumes, we see that they're limited in tryptophan and methionine, but if we combine them with grains that are limited in lysine, isoleucine, and threonine, but not in tryptophan or methionine, then we get this complementary food in which we're getting all of our essential amino acids. So here's our animal foods that contain all of our essential amino acids, and here we can see that we would want to pair legumes with grains or with nuts or seeds in order to have a complementary food. And then we can also classify protein by how they are metabolized. So if we have a dietary surplus of amino acids, the nitrogen of the amino acids are eliminated, and we'll talk more about that moving forward, but the carboskeleton of the amino acid is conserved. And through this, we can produce carbohydrate through gluconeogenesis, or we can produce fatty acids through fatty acid synthesis pathways. So depending on the amino acid, we would either be able to make the carbohydrate or the fatty acid or both. So amino acids that are able to be degraded and then go through gluconeogenesis to produce glucose are called glucogenic. So glucogenic amino acids give rise to either pyruvate or some TCA cycle intermediate, and these serve as precursors to for gluconeogenesis. And all amino acids except lysine and leucine are at least partly glucogenic. So lysine and leucine are exclusively ketogenic. So they give rise to only acetyl-CoA or acetoacetyl-CoA. And then there are also amino acids that are both glucogenic and ketogenic. Okay, so let's move on to the functions of proteins. So the functions of proteins are very, very diverse and have really the importance of proteins in our biological functions cannot be underestimated. So we're going to talk about some of the main categories of functions for proteins. And we can see here their growth and maintenance as enzymes, hormones, antibodies. They help in fluid balance and acid-base balance. They serve as transporters. They provide us with energy, serve as lubricants, and have other functions as well. Okay. So growth and maintenance. So we need to ensure availability of amino acids to build proteins of new tissues. So periods of growth in the life cycle include the embryo when it's developing, and of course growth throughout childhood. So if a child is not, well, I mean anyone, but especially these occur in children, if a child is not intaking enough protein to meet their needs for growth, we have what's called protein energy malnutrition. And this the kwashiorkor specifically often affects first child if we're in like maybe a non-industrialized country where there's not as many resources. The kwashiorkor will affect a first child when a second child is born. So when a second child, often, this is kind of a pattern, I guess I should say, not always, but it would be second child may be of higher risk, or the first child may be of higher risk because the second child comes along, and so mom stops breastfeeding the first child and starts breastfeeding the second child, and therefore the first child is cut off from a really quality source of protein, maybe in a setting where there aren't as many quality sources of protein. So other needs for growth and maintenance would occur when we need to synthesize blood that's lost when we have an injury, such as a surgery or a burn or a hemorrhage, also to heal wounds. We need protein for just regular hair and nail growth, and for maintenance of our bone, teeth, ligaments, and tendons, and arteries, because we need collagen. So the cells, so amino acids are also needed to replace worn out cells. So the cells of the digestive tract are constantly being shed and excreted, and we need adequate protein intake to synthesize new cells. Also, our skin cells die and rub off and are replaced from underneath every day, so we need amino acids to replace those cells as well. So for all new growth, amino acids must be constantly resupplied by food. And then, sorry, I should point out with this picture here, for the protein energy malnutrition, we'll see moving forward that that protein is needed for fluid balance. And so when children who have this kwashiorkor don't have enough protein, they're not able to balance their fluid as well. And that's why we kind of see these bloated bellies on these very malnourished children, because they don't have the protein integrity on a cellular level to regulate their fluid balance. Okay, proteins are also used as enzymes, and all enzymes are proteins. So as we've seen, most proteins are designated with an ACE suffix, and there are thousands of different kinds of enzymes that are specific for different chemical reactions. We've talked a lot about enzymes needed in digestion and release of energy from body stores, and we also have an extensive need for enzymes in tissue growth and repair. So enzymes are specific to a particular reaction, and the name of the enzyme often indicates the function of the enzyme. So we've seen in our discussions, for instance, the kinase enzymes always add a phosphate group on, whereas the phosphatase enzymes take a phosphate group off. So here's some more examples of the specific functions of different kinds of enzymes. So we know that hydrolases cleave enzymes, whereas ligases or synthetases join compounds together. And then some enzymes do require cofactors or coenzymes needed for carrying out their reactions. So even though we might have enough proteins to produce these enzymes so that they function, they won't function if they don't have their coenzymes, such as they could be either a mineral or a vitamin, like a B vitamin. Okay, proteins also serve as hormones, though not all hormones are made of proteins. And hormones do not catalyze enzyme or chemical reactions directly like enzymes do, but instead they act as messengers that maintain a normal environment in the body. So hormones regulate overall body conditions. So for example, insulin and glucagon regulate blood glucose, calcitonin and parathyroid hormone regulate blood calcium. So they keep our body in a state of homeostasis. And then here's some other examples. So the amino acid tyrosine is used to synthesize catecholamines. Melatonin is derived from in the brain from tryptophan, and other peptide hormones, including the adrenocorticosteroid hormone, our growth hormones, and our antidiuretic hormones are also peptide hormones. Proteins can also serve as antibodies, and this is just an excellent example of how proteins are not specific for one organism, but really can be transferred, have similarities between organisms. So antibodies are formed in response to the presence of antigens that invade the body. So when this foreign substance or antigen invades the body, the antibodies are manufactured to inactivate them. And antigens, they are either foreign proteins or they're other large molecules. So they can either be a bacteria or a virus or a toxin, or they could be a specific protein in a food that may not be an antigen for all people, but could be an antigen for a specific person in the form of an allergy. So once the body has learned to make a particular antibody in response to a specific antigen, it never forgets. And this is what is called our immunity. This is what happens when we get our vaccines, for instance. We learn to make antibodies, and our body remembers that. So so the body can't maintain a disease resistance if it doesn't have the proteins for antibodies. So malnutrition, however, does impair the immune system because without adequate protein in the diet, the immune system can't make these specialized cells, these antibodies, or the other tools that it needs to function optimally. And we often see protein deficiency and immune incompetence that occur together. So here's just a real short YouTube video that'll kind of brush us up on how antibodies work, the proteins that our antibodies work. Oops. Okay, well, I'll let you watch that on your own. It's just 1 minute, just to kind of give you a brush up. But hopefully we'll be able to access the internet. The other ones. Actually, I'm going to pause this and make sure I'm connected to the internet. Just 1 second. Okay, let's try this again.
A foreign bacteria has invaded. One way the body protects itself is with small proteins called antibodies. These Y-shaped molecules recognize the intruder and bind to its surface at special sites called epitopes. When antibodies begin to cover the bacteria, it is called opsonization. This process attracts a phagocyte, a large cell that digests unwanted particles in the body. The phagocyte binds to the antibodies and engulfs the bacteria. The microbe is then killed and digested, and the body saved.
Okay, so moving on. Okay, proteins are also important in fluid balance. So proteins help regulate the quantity of fluids in cell compartments to maintain fluid balance. So we need fluid balance in our cells because if there's too much fluid in the cell ruptures, and if there's too little fluid in the cell, then it's unable to function. So the method through which proteins help to maintain this fluid balance is that proteins attract water, but they can't diffuse freely through the cell membranes like water can. So therefore, the water attracting protein stores inside the cell help bring the water into the cell and keep it in the cell instead of diffusing back out of the cell. And this same method occurs in first proteins secreted into the blood vessels to maintain blood volume. So you can see just in this little graphic here, the pressure of pumping blood pushes fluids out of the blood vessel, but the proteins in the blood vessel and capillaries help to attract water back into the blood vessel in order to kind of keep a constant volume. Okay, proteins are also needed for acid-base balance. So as we've seen demonstrated time and time again, through normal physiologic processes, we produce acid and bases, and these have to be carried by the blood to the organs of excretion in order to avoid going outside of our very narrow range of what is okay for our blood, or our normal pH for our blood, which is very essential for physiological function. So the blood has to maintain its own acid-base balance because we constantly have products that are acids or bases entering the blood, and these are very tightly controlled. And so if our pH goes off too much from our blood, then we're either considered to be in acidosis or alkalosis, which can lead to coma or death. So some proteins are able to act as buffers to maintain normal pH. That is, they ameliorate any change in pH. So some examples of this would be proteins that we find in red blood cells or in muscles that are able to pick up excess hydrogen from the blood if we have too many acids in our blood, and able to release this hydrogen when our blood is kind of tipping towards a little bit more basic. And then one function that we're all very familiar with are the function of proteins as transporters. So these proteins specialize in moving nutrients and other molecules in and out of cells, and can be switched on or off in response to hormonal stimuli. So we can see in this little graphic here, yes, we can have passive diffusion of our of certain molecules through a cell membrane, but even with facilitated diffusion and of course active transport, we need membrane proteins in order to help us maintain good physiologically functioning concentrations of these nutrients in and out of the cell, and it's these transport proteins that help regulate the concentration of these molecules. So if facilitated diffusion, we do need these transport proteins to get the molecules in and out of the cell, but they still move with the concentration gradient. They're still moving from areas of high concentration to low concentration. Whereas in active transport, our transport protein is actually using ATP to move molecules from lower concentration to a higher concentration. And so this is the type of transport protein that we've talked about the most, but other transport proteins actually move nutrients and molecules throughout the body fluids from one organ to another. And here are some examples of transport proteins. So an important one, of course, is hemoglobin, which carries oxygen in our blood. And we also have lipoproteins, which transfer lipids around the body, like we've talked about. Some other proteins that we can consider as transport proteins would be albumin, which transports calcium and zinc and B6. And a couple of other examples are noted here too. And then, of course, proteins can be utilized as energy. So our body's first, preference is to use carbohydrates as energy, and then if carbohydrates aren't available, to use fat as energy. However, if we don't have enough carbohydrates or fat to use as energy, then we can use proteins for energy. So we must have enough calories from carbohydrate and fat in order to provide our body with adequate energy before amino acids can be used for their most important function, which is making proteins. So if our bodies don't have enough energy from carbohydrates or fat, then they'll go and use amino acids for energy, and in that way the amino acids will be deferred from their responsibilities or function as making the proteins for all the functions that we've talked about until now. So amino acids are first broken down. The amine group is taken off and incorporated into urea and then excreted through the kidneys. And then the carbon, hydrogen, and oxygen, what we would call the carbon skeleton, are available for immediate use as energy. So, so not only are we going to utilize amino acids for energy if we don't have enough carbohydrate and fat, but we also may utilize them for energy if we just have a lot of amino acids. So think of a person who is getting 35% of their calories from proteins. So we're gonna, at that point, we're going to use some of these amino acids for energy. So again, the amine group is removed and excreted, and then the carbon skeleton is converted to glucose, which could be stored as glycogen, or fat. Now, if we don't have enough amino acids, if we don't have enough amino acids for our kind of basic physiologic functions or for energy use, then our tissues are broken down to provide these amino acids that are used to maintain our basic physiologic functions, such as our heart and our lungs and our brain. Okay, and then proteins can be used as lubricants, such as the case with our mucoproteins that we see that are a component of our mucus. And there's also proteins that surround our joints and help facilitate the movement of our joints. And then finally, proteins can be used. Another function of proteins is that they participate in blood clotting, as in the molecule fibrin, and also in our vision. Oh, and I should say the mucoprotein that we find in mucus is a conjugated protein that contains hexosamine. So we also have non-protein pathways of amino acid nitrogen utilization. So amino acids that we get from the diet are primarily either synthesized into proteins that we need for the functions we've discussed, or they're used for energy. However, because they are our main source of nitrogen, they're also involved in the synthesis of other nitrogenous compounds that are important for physiologic function. So glycine, for example, is involved in 6 synthetic pathways that we see here. So it's involved in the synthesis of creatine that we need for muscle metabolism, heme that we need for oxygen transport and oxidative phosphorylation, glutathione for our protective reactions, and so on. And of course for our nucleic acid bases. Okay, so that brings us to deamination and transamination. Now, to utilize amino acids for metabolism, we have to remove the amino group from the amino acid by either deamination or transamination. So if the amino acid is needed for energy or used to make fat, it first must be deaminated. So after we take off the amino group, the remaining molecule is no longer an amino acid because it needs an amino group to be an amino acid. So at that point, it's called a carbon skeleton or an alpha keto acid. And these carbon skeletons can be catalyzed in a variety of ways, which we touched on a little bit earlier. So the amino acids can, some amino acids can be converted to pyruvate for energy production or for gluconeogenesis. Some are converted to acetyl-CoA for energy production, ketone body production, or synthesis of cholesterol and fatty acids. Or some products of amino acid degradation can enter the TCA cycle directly and therefore be utilized for energy. Okay, so in deamination, we are taking the amino group off and excreting it. Okay, so when we take so here we have our amino acid, and then we take our amino group off, and we are left with the amino group on its own, and then also, sorry, this corrected itself, but this should be alpha keto acid. So this is the alpha keto acid or carbon skeleton, which is basically the amino acid without the amino group. And the enzymes that catalyze the deamination are often called deaminases, but not always. Now, transamination occurs when the body transfers the amino group from the amino acid from an amino acid onto a new carbon skeleton, or alpha keto acid, to form a new non-essential amino acid and a keto acid. And this occurs in the liver. So here's kind of a little graphic that shows, so we have one substrate that's an amino acid, and the amino group is transferred onto this keto acid. So we're left with a new keto acid and then a new amino acid with, which was the keto acid with the amino group from this original amino acid. So one place that we see, so through many reactions involving many keto acids, the liver can synthesize new non-essential amino acids. And they're called non-essential because we can synthesize them in our liver. So one example that we've talked about in this class is the pyruvate alanine cycle. So in the muscle, we have alanine, we have the conversion of pyruvate to alanine through transamination with a different amino acid. And then the alanine goes to the liver and is deaminated or transaminated. The amino group is taken off, and then it forms pyruvate, which can then be used for energy production, and forms a new amino acid, which is glutamate. Okay, so let's go specifically into how amino acids are converted to different substrates. So amino acids can be converted, some amino acids, I should say, can be converted to pyruvate that can then be converted to glucose via gluconeogenesis. So amino acids that are used to make acetyl-CoA, such as our strictly ketogenic amino acids, can provide energy or make body fat, but they can't make glucose. And as we know, we need some glucose for tissues that can really only utilize glucose, such as our red blood cells and our central nervous system. So, and then amino acids can enter, once they're degraded, they can have products that can enter the TCA cycle directly and continue to produce energy. Okay, and here's a nice graphic that shows where different amino acids can be degraded. Once specific amino acids are degraded, where they can enter into this pathway, into this oxidative pathway. So here are alanine, cysteine, and tryptophan, as well as serine that can enter as pyruvate. We have our branched chain amino acids that can enter at acetyl-CoA. And then you can see just a bunch of other amino acids that can be degraded to enter at specific points in the TCA cycle, at which point they would be oxidized and produce energy. Okay, and as we've talked about, some amino acids can be converted to acetyl-CoA. We saw that in this graphic here, all of these. And if energy is not needed, or we have excessive energy for our needs, then these fatty acids that were produced from the acetyl-CoA can be stored in the form of triglycerides in adipose tissue. So leucine is, oops, what happened there? Sorry about that, just a second. So leucine is the only amino acid whose catabolism generates HMG-CoA, which we know we need for cholesterol synthesis. However, other amino acids can generate acetyl-CoA, which can then be metabolized in the liver for cholesterol. So when energy intake is greater than need, and protein intake is high, then the amino acids can be degraded and stored as fat. So in order to be converted to amino acids that we need in new products, the body is continually undergoing deamination. And therefore, we have these extra amino acid or amino groups, which are nitrogenous in nature. And really, these ammonia groups that are formed when we take these amino groups off of the amino acid are toxic. So the liver detoxifies the ammonium group by combining it with carbon dioxide to produce urea, and then it's entered into the bloodstream. And the urea circulates until it passes through the kidneys. At that point, it's excreted in the urine. So if our liver isn't functioning properly, then the liver is not able to detoxify the ammonia, and we'll have high levels of blood ammonia. And we see this a lot with like cirrhosis or hepatitis. And if we have kidney disease, then the kidney is not able to adequately filter out the urea for excretion in the urine, and therefore we have high levels of blood urea. So if we are consuming a very high protein diet, then we're going to have more deamination, more production of ammonia groups, and therefore we really need to drink more water in order to keep the urea that we produced as a kind of a product of the ammonia detoxification. We need to drink more water to keep more of this urea into solution, or else urea will begin to accumulate in the blood. Okay, and just to talk on a therapeutic use of amino acid, just to touch on one, there's a chemotherapy drug, asparaginase, that's used to treat acute lymphoblastic leukemia. And this asparaginase breaks down extracellular asparagine, which is required for cell survival. So we know that we need certain amino acids for cell growth and survival. So here we have our graphic. So we have our asparagine, and we have the that the amino acid is needed for growth of these tumor cells, so then they grow and they reproduce. But the asparaginase breaks down the asparagine, so then what happens is with the tumor cells, they can't grow and reproduce. So our normal cells can synthesize asparagine, but some malignant cells could not. So if we introduce this asparaginase to break down asparagine, then we effectively cut out the tools that the tumor cell needs to grow. Okay, so here's a summary for the section that we just talked about. So proteins are a very broad class of macronutrients with many different types of physiological functions. They're very diverse, and so they have different methods by which they're classified. And to utilize amino acids for metabolism, we must remove the amino group by either deamination or transamination, and then the amino acids can be used to produce a variety of other molecules such as glucose or fatty acids, and these can also be storage forms of energy. And then the amino group that is removed by deamination must also be excreted in order to avoid toxicity. And here are our practice questions, so I'll let you do those on your own. And let's move into our next section, which is evaluation of protein quality and amino acid availability from the diet. So our objectives are to define protein quality and indicate methods of measuring protein quality, understand and calculate amino acid chemical scores, be familiar with BV, PER, and PDCAAS, which we will talk about, explain nitrogen balance, and evaluate amino acid availability from the diet under different conditions. So the quality of the protein is expressed as its ability to provide nitrogen and amino acids that are required for maintenance, growth, and repair. So the quality of the protein is determined by the amino acid composition of the food, as well as the digestibility of the food. So there is a variety of methods that we can use to measure protein quality. So, and really the quality rating of the food depends on which method is used. And all methods have their strengths and limitations. So we can use a chemical score, biological value, protein efficiency ratio, or a protein digestibility corrected amino acid score. Okay. So for the amino acid score, or also called the chemical score, this test compares the test protein's essential amino acid pattern to a reference protein. And this is often an egg protein, which is contains all of our essential amino acids. So the essential amino acid that's present in the lowest quantity is defined as the first limiting amino acid, and then the second most limited essential amino acid is considered the second limiting amino acid. So generally, the limiting amino acids will determine how well or how poorly the given protein is able to be used in the body. So measures really its completeness. And this test is done with an amino acid analyzer or a high-performance liquid chromatography. Okay, and when we look at the outcomes of the different scores for these methods, we see that supplementation of limiting amino acids or combining proteins with different limiting amino acids could increase protein quality. Makes sense. Okay, and this is the actual equation for which we calculate chemical score. So the content of each of the essential amino acids in the food protein compared to the content of these same essential amino acids in a reference protein, and then we multiply it by 100 to get a percentile. So here's an example here. So if the content of lysine in a test protein is 2.5%, and the ideal level of protein is 5.5%, that is that in the reference protein, then the amino acid score is 2.5 divided by 5.5, or 45%. And the higher the percentage, the better the protein. However, this method does not take into account digestibility, which is a major limitation. And so we rarely rely on this measure for protein quality only to rate protein. It might be part of like a profile by which we look at protein quality, or just one part of it. And then we have the measurement of biological value. So this is given as the amount of nitrogen that's retained in the body for maintenance or growth, and divided by the amount of nitrogen that's absorbed from that protein. So how this test is conducted is that a subject is fed nitrogen, a nitrogen-free diet for 7 to 10 days, and then they're fed a diet containing the test protein for an equal amount of time. And then the researchers measure the nitrogen that's excreted primarily in urine, but also in feces. And the change in the urinary or fecal nitrogen excreted between the two diets is calculated. So the more nitrogen that's retained, the higher the protein quality. And this method is nice because it does take digestibility into account, or how much is absorbed into account. So a BV of 100 indicates that there's complete utilization of a given dietary protein. None was lost. Okay, then we have our protein efficiency ratio, which basically measures how well a given protein supports weight gain divided by the quantity of the protein consumed. And this is used particularly with infants. So how much weight was gained for every gram of protein ingested. And then we have our gold standard, which is our protein digestibility corrected amino acid score. So this is the ideal scale to rate proteins for their ability to meet human requirements, because it goes beyond the chemical score by factoring in the digestibility of a given protein, giving the amino acid profile more relevance to human needs. And because of this, it's recommended by the World Health Organization and the FAO. So the way that this score is determined is by comparing the chemical score of a food protein with the amino acid requirement for children and adults, and then correcting it for true digestibility of the protein. So we see that wheat, which is lysine limiting, has a score of 44, and chickpeas that are also limiting, have limited essential amino acids, has a score of 87%. But a diet that has a mixture of wheat and chickpeas and skim milk meets a max score of 100. And then this isn't super clear, but here's just the PDCAS scores of selected foods. So we can see, as we would anticipate, our animal proteins have a higher score, whereas our plant proteins, which have some missing essential amino acids, on their own they would have a lower score. But if they were combined, then they might have, then they would have a higher score. Okay, so next for just a minute, I want to talk about nitrogen balance. So, like we talked about, we excrete nitrogen in the form of urea in our urine. However, we also lose nitrogen through other methods, such as in feces, sweat, skin, nails, hair, and other bodily excretions. But since it's difficult to measure all of the routes of nitrogen loss from the body, we usually estimate losses from all other sources other than urine, and it's urine that we actually measure. So nitrogen balance can either be positive, negative, or in equilibrium. So if we have a positive nitrogen balance, then the individual is consuming more than they need. They're consuming more than they're losing and storing the nitrogen in the body. And this occurs in periods of growth, such as infancy, pregnancy. We also see it in times of lactation, or if someone is needing to heal, such as if they have burns or severe illness. Nitrogen balance can also be negative, which means the individual is losing more nitrogen than they're consuming, and this indicates that body protein is being lost. And we see this in restrictive weight loss diets, such as those with very, very low calories, like less than 800 calories. We would see this in maybe anorexia nervosa or in patients who have cancer. Or our nitrogen balance can be in equilibrium. So we're neither storing nor losing body nitrogen. And this just happens in a healthy, regular adult that's not pregnant or lactating and who is consuming a balanced diet. So nitrogen balance depends heavily on caloric intake. So an individual who's fasting is going to lose a lot of nitrogen, whereas individuals who consume a high-protein diet are going to excrete more nitrogen. So in urine, the nitrogen is mainly in the form of creatinine, amino acids, ammonia, and uric acid, whereas in the feces, it's mainly in the form of amino acids and ammonia. And urinary urea nitrogen tests are used to determine a patient's nitrogen balance. So if they have a negative nitrogen balance, that really indicates that the patient needs to consume more dietary protein. Okay, so amino acid availability from the diet can be altered according to a couple of different conditions. So the first that I want to discuss is food processing. So 4 types of damage to proteins can occur in food processing. So the first one is if there are mild heating conditions. So one example is that in the Maillard or Browning reaction that we've all learned about, I'm sure, that can result in loss of lysine. We also know that lactose interacts with lysine residues to render them unavailable. So if so lysine could be lost in the presence of processing a product with milk. We also know that damage to amino acids can occur in severe heating conditions. So proteins can be resistant to digestion and their availability becomes reduced. Damage to amino acids and therefore decreased availability can also occur from severe treatment of the amino acid with an alkali product or under oxidation. So, for instance, sulfur dioxides, when they're added to foods, which I don't know about you, but I see those in my foods or ingredient lists, I guess, at the grocery store all the time. I see those in dried fruit a lot, which is always discouraging. But these can result in the loss of methionine. Now, amino acid availability can also be altered by consuming excess amino acids. So we can actually have toxicity of specific amino acids, and the most toxic are methionine and tyrosine. And threonine in very large quantities can also cause moderate growth reduction. So we can also have, when we have excessive intake of a specific amino acid, we can have amino acid antagonism. So this is when we have excess of a specific amino acid, which causes a depression in growth rate, which is alleviated by the addition of a structurally similar amino acid. So what was happening was because we consumed a specific one, it antagonized digestion and incorporation of structurally similar amino acids. And another result of excessive intake can also be amino acid imbalance. And this occurs, we know we have an amino acid imbalance when we change the proportion of the amino acids that are causing our growth failure, and then it's alleviated by adding one or more of the limiting essential amino acids in the diet. However, this particular issue is unlikely to occur in humans who have a balanced intake. Okay, so here's a summary from this section. So quality of source of protein, which is nitrogen. So I guess, hold on, I should go back a little bit and say that when we're measuring the quality of the source of protein, we're really measuring go back and just see exactly how I wanted to say that. We're really measuring the what we're really measuring is analytically is the nitrogen. So the quality of the source of protein is an expression of its ability to provide nitrogen and the amino acids that are required for growth and maintenance and repair. Now we have a lot of different methods of measuring protein quality, and each has their advantages and disadvantages, but the most accurate method is the PDCAAS method, which expands on the chemical score and factors in digestibility, which give us a really relevant score for human need. And we also saw that food processing and overconsumption of a single amino acid can impact bioavailability. And here are your practice questions. So I'm going to move on to our next section. And our last section that we're going to talk about today is digestion and absorption of protein. So our objectives are to understand protein digestion, explain the role of different enzymes in digestion, understand protein absorption and transport, and discuss physiological conditions that impact protein synthesis. So our main goal with digestion is to degrade the protein food that we consume into individual amino acids to be delivered to the body cells. And in the body cells, these amino acids are put back together in order to produce needed proteins. So in our mouth, just kind of like our other macronutrients, we don't really have too much meaningful digestion. Our main function of our mouth and digestion is to chew and crush and moisten these protein-rich foods to mix them with saliva so that they can be swallowed. And then our serious digestion of protein begins in the stomach with the action of hydrochloric acid, which uncoils protein strands and also activates enzymes in the stomach. So when we have our protein enter our stomach, this activates the enzyme pepsin and hydrochloric acid, which hydrolyzes the proteins into smaller polypeptides. Now, hydrochloric acid is stimulated by the hormone gastrin, also gastric releasing peptide, and the neurotransmitter acetylcholine. So hydrochloric acid, as we said, it denatures the protein structure. So proteins are in these kind of really tight strands that are all kind of bunched up together, and the hydrochloric acid helps to kind of uncoil these strands in order to give the enzymes access to these polypeptide chains. And hydrochloric acid also activates the enzyme pepsinogen into its active form, pepsin. Okay, so pepsin cleaves proteins into smaller peptides and free amino acids. And pepsin also inhibits pepsinogen synthesis and functions at a low pH, which we know occurs in the stomach because of the hydrochloric acid. At this low pH, the pepsin functions as an endopeptidase, which means it attacks peptide bonds within the polypeptide chain, not just at the end. So the end product is of degrading these proteins is that we have the shorter polypeptides in our acid chyme, and the chyme then goes through the pyloric sphincter and into the duodenum, where it will be further digested. So we can see our nice graphic here. We have our long polypeptide chain, and then we have the action of pepsin, which breaks it into smaller polypeptide fragments. But in order for pepsin to be activated, we have to have the stimulation of pepsinogen, and then we have to have hydrochloric acid in order to activate pepsinogen into its active, or convert pepsinogen into its active form, pepsin, which is then going to break down this polypeptide chain. Okay, so then we move into the small intestine, where digestion continues until almost all pieces of the protein are broken into dipeptides, tripeptides, and more free amino acids. So the acid chyme that moves into the duodenum stimulates the hormone cholecystokinin and secretin, and then these are released from the mucosal endocrine cells in our small intestine. These hormones are carried by the blood to the pancreas, which then is stimulated to secrete alkaline pancreatic juice, which will neutralize the acidic chyme, and it also stimulates the pancreas to release digestive proenzymes. So these enzymes that we get from the pancreas that go into the small intestine, and the enzymes from the small intestine itself, help to split the polypeptides further. So we have these polypeptides, we have our proteases from our pancreas and small intestine, and then these are broken down into dipeptides, tripeptides, and free amino acids. Then these molecules here go to the surface of the small intestine, where enzymes on the surface of the brush border hydrolyze the peptides further down to individual amino acids so that they can be absorbed into the mucosal cell. So we have our peptides, and then they are degraded by dipeptidases and tripeptidases at the brush border to break them down to amino acids that we can absorb into the cell. Okay, so the digestive proenzymes, which we also call zymogens, are secreted by the pancreas. And these are inactive forms of the enzyme. Okay, so secretion of these proenzymes are, again, stimulated by cholecystokinin, and our main enzymes of action to degrade proteins or peptide chains are trypsinogen, chymotrypsinogen, procarboxypeptidase A and B, proelastase, and collagenase. So within the small intestine, these proenzymes that were secreted from the pancreas need to be activated so that they can help break down these peptides. So then when the proenzyme trypsinogen enters the small intestine, then we use the enzyme enteropeptidase or enterokinase to convert trypsinogen to trypsin, which is the active form of the enzyme. So this enteropeptidase is secreted at the brush border of the small intestine in response to cholecystokinin and secretin hormones. I'm going to put the correct one. Don't worry, I'll post the correct one. But I just want to make sure I don't forget some of these. Okay, so then our digestion continues in the small intestine. There's a lot that's done in the small intestine here. So once the trypsin is formed from its inactive form, there's a cascade of activation of other enzymes. So the trypsin helps serves as an enzyme for conversion of other proenzymes to their active forms. So it can serves as the enzyme for the conversion of chymotrypsinogen to chymotrypsin, or chymotrypsin, I should say, procarboxypeptidases to carboxypeptidases, and proelastase to elastase. And all of these products here are also endopeptidases, or active digestive enzymes. So other peptidases that we find at the brush border include aminopeptidases, dipeptidyl aminopeptidases, and tripeptidases. Okay, so after we go through all of these reactions, our final product are dipeptides, tripeptides, and free amino acids. And these can be absorbed across the brush border of the intestinal epithelium mucosal cell. So I just kind of want to go into these enzymes with a little bit more detail. So trypsin is a product of trypsinogen, and it inhibits trypsinogen synthesis. It cleaves the peptide bonds next to the amino acids lysine and arginine, and it helps convert other proenzymes to their active form. Chymotrypsin cleaves the peptide bonds next to the amino acids phenylalanine and other amino acids here, methionine, tyrosine, histidine, etc. The carboxypeptidases A and B cleave amino acids from the carboxyl end of the polypeptides, and they also depend on zinc in order to function. Carboxypeptidase A hydrolyzes the terminal aromatic or branch chain amino acids, and carboxypeptidase B hydrolyzes terminal arginine or lysine. Elastase and collagenase cleave polypeptides into smaller polypeptides and tripeptides, and aminopeptidase cleaves amino acids from the amino end of the dipeptide. And then just a couple more, the dipeptidyl aminopeptidases cleave the essential amino acids from amino end of the peptides, and also depend on magnesium as a cofactor. And tripeptidases cleave tripeptides to dipeptides and free amino acids, as we would expect. Okay, so here's a nice graphic here that's showing how this process occurs at the brush border and how it's absorbed into the epithelial cell and eventually into the bloodstream. So we see we have our amino acid protein in peptide fragments here. We have our brush border enzymes here and our microvilli that are here. So first, in this first stage, our protein fragments are digested by pancreatic proteases and by brush border enzymes such as carboxypeptidase, aminopeptidase, and dipeptidase on these mucosal cells here. Then once it's in the epithelial cell, the amino acids are absorbed by active transport into the absorptive cell and move to the opposite side of the cell. And then finally, they leave the small intestinal epithelial cell by facilitated diffusion and enter the capillary to enter the bloodstream, where they're carried to the liver. Okay, and here's a little bit drier graphic, but it gives us a really good picture of all of the enzymes that are in play. So this is just to kind of recap what we've talked about. Food enters the stomach, gastric mucosa are stimulated, gastrin is released, hydrochloric acid is secreted, and pepsinogen is released. Hydrochloric acid helps to denature the proteins and also converts pepsinogen to the active form of pepsin, which helps to hydrolyze some of the peptide bonds. Smaller peptides are then released into the intestine, which stimulates the release of CCK, or cholecystokinin, and also the release of secretin. Secretin helps to neutralize our very acidic chyme. Cholecystokinin causes secretion of trypsinogen, which is our proenzyme from our pancreas. And then the enteropeptidase helps to convert the trypsinogen to the active form trypsin. Trypsin helps to convert other proenzymes to their active form, and these enzymes are used to hydrolyze the proteins into smaller peptides. So these, let's see here. So then we have hydrolysis of the carboxyl terminal end and of the amino terminal end, and eventually we get our very small peptides and our free amino acids, which then are absorbed into the blood and go to the liver. And here we have another slide to sum it up. It's kind of a little blurry, but let's go through it anyway. Gastric cells release gastrin, causing release of HCl. HCl denatures proteins and converts pepsinogen to pepsin, which begins to digest the proteins. So we're all still in the stomach here. Then the partially digested proteins enter the small intestine, causing release of the hormones secretin and CCK. Then these hormones stimulate release of pancreatic proenzymes and bicarbonate to neutralize the chyme. Then the pancreatic proenzymes are converted into their active enzymes in the small intestine, which help to digest the polypeptides into tripeptides, dipeptides, and free amino acids. And then intestinal enzymes at the brush border complete protein digestion so that they can be absorbed. And then I won't go through this, but here's just kind of a nice summary of important players enzymatically in the digestion of proteins. Okay, so di and tripeptides are broken down into individual amino acids as they cross into the blood through the wall of the intestine. And here they form pools of free amino acids. Now, the blood ultimately will distribute these free amino acids to tissues, especially the muscle, where they can be built back into proteins. So we took the proteins apart, and then we can put them back together in a way to synthesize the proteins that we need in our body. So we can take the proteins we consume and make them into the proteins that we need. So all amino acids in the blood are free, and they're taken to the liver, where one of three things happens. So first, they might form part of an amino acid pool in the liver. And you can just kind of think of this as a reserve of the specific amino acids that we might need to build specific proteins. These free amino acids can also be used at that time in the liver to synthesize proteins in the liver, or be made into albumin or other proteins that are exported into the blood. So, in addition, if we have excessive amino acids, then we can start to break these down in the liver in order to use the carbon portion to make glucose or fatty acids, and then we excrete the amino group in the form of urea. So if we do produce glucose or fatty acids, we could also build these into glycogen or triglycerides for storage. Okay, so we have free amino acids in the blood. They go right to the liver. They're going to form an amino acid pool. They're going to be used to synthesize proteins, which will either stay in the liver or be exported in the blood, or they're going to be catabolized for energy or storage of energy. And we have a nice graphic here that just kind of shows everything that we've been talking about. So we have amino acids in the liver. They can be released into the blood for tissue proteins. They can be used to synthesize nucleotides, hormones, or protoporphyrins. Or they can be catabolized, in which case the ammonia group is excreted as nitrogen, or but also they can be used to either be degraded as energy or to build glucose via gluconeogenesis and then ultimately glycogen, or they can be used to form acetyl-CoA, which will be synthesized into fatty acids or triglycerides, or the intermediates can go into the citric acid cycle to be utilized for energy via oxidative phosphorylation. So just to kind of sum up what we've talked about, for absorption and transport, we've got 7 steps. We've got absorption of free amino acids and peptides after they're digested. They're then taken up by the liver. They're synthesized into proteins, especially albumin, that can be exported in the blood, or they're catabolized if there's excess amino acids. And then the amino acids can be distributed to the rest of the body, especially the muscle, where they're taken up and built into new proteins. And this also occurs in the for various other body tissues, such as the pancreas and our epithelial cells. And then we have excretion of amino acid nitrogen, primarily through the urea. Okay, so we absorb a lot, or a vast majority of the protein that we take in. So we have, and one, I guess, handy mechanism is that we're able to recycle our proteins in our bodies. So we have so many functions that our proteins serve, and our bodies have evolved in order to really conserve these amino acids so that we can always have them on hand when we need them. So a couple of examples of how this occurs is with our digestive enzymes. So we use our amino acids to build our proteins into digestive enzymes, but then once these are secreted into the digestive tract, we degrade the enzymes, and then we're able to reabsorb these amino acids from that degradation. In addition, we roughly slough off about 1/4 of the cell linings of our GI tract each day. So with this sloughing off of cells, we again can degrade these cells and then reabsorb the proteins. So as we said, when free amino acids enter the portal blood for distribution, they go to the liver, and this is the major processing site for any amino acids. And the liver and the muscle are the major organs that are involved in the degradation of excess amino acids. However, the liver is the main and only site for degradation of most essential amino acids, with the exception of the branched chain amino acids, where catabolism occurs mostly in the muscle. So protein synthesis in the liver and muscle are very sensitive to protein influx. So after a protein-containing meal, there's a period of increased synthesis. And this is mediated by insulin secreted in response to amino acid or glucose influx from the meal. So insulin enhances the transport of these amino acids and also has an important effect on protein synthesis. So we know if we have insulin, it's saying, I've got extra, let's start to store these or use them. So muscle protein is the largest reserve for which amino acids can be drawn from in times of need. And these include if someone is fasting or starving, or if a substantial portion of their amino acids are being utilized for gluconeogenesis. So after a meal, the influx in amino acids results in an increase in protein synthesis in the liver and the muscle, and including albumin synthesis, and also degradation of excess amino acids into a glucose precursor or acetyl-CoA for fuel or storage in the form of glycogen or triglycerides. So again, this makes sense. We have excess amino acids. We're either going to store them or use them by building proteins or breaking them down to use for fuel. As the absorption of the amino acids in the intestine declines after a meal, so we're kind of increasing the time since our meal, then the protein synthesis slows down, but degradation does continue. So this can result in a net release of amino acids, especially from the liver, as fasting begins and progresses. However, our bodies are incredibly amazing and very adaptable, so that if there's long-term fasting, there's adaptation in how we metabolize protein and amino acids, and this results in reduced need for breaking down muscle. So after a while, our bodies are able to actually decrease this muscle breakdown. Okay, so let's watch a little summary of protein digestion. It's kind of a simplistic one, but it's still nice to get an overview.
Breast cancer doesn't just affect one person.
Did you make a wish?
It impacts everyone.
Proteins are important nutrients that your body uses to build cellular structures that carry out vital functions. In fact, the basic building blocks of proteins are so important to your cells that if you did not consume protein, it would be like trying to build a house without tools or nails. In this lesson, you will learn how proteins are broken down into their basic units called amino acids. You will then see how amino acids are absorbed through the small intestine wall and used by your body to build cellular structures and components. Now, when we talk about digestion, we look at protein as a large molecule composed of one or more chains of amino acids. When you feel hungry, you typically think of protein as a food such as eggs, milk, meat, nuts, or beans. Regardless of which way you want to look at protein, one fact remains, and that is amino acids are the basic building blocks of protein. Now, the term building block is a good way to describe amino acids because they are literally used by your body for building biological substances. In fact, many of these biological substances are actually specialized proteins. So you might eat an egg for breakfast, and once you swallow it, enzymes break down the protein in the egg into amino acids. Then those free amino acids recombine in different ways to form specialized proteins. These specialized proteins become different things such as enzymes or antibodies or hormones, or they might become structural proteins such as muscle proteins or collagen found in connective tissue. Now, it's good to keep in mind that protein digestion is not as simple as eating an egg and magically getting amino acids. A large protein molecule breaks down via a few intermediate steps in the stomach and in the small intestine before it becomes the tiny amino acids. So let's take a look at how proteins are broken down by your digestive system. So protein digestion begins in the stomach with the action of an enzyme that we previously learned about called pepsin. Pepsin is the active protein digesting enzyme of the stomach. When pepsin acts on the protein molecule, it breaks the bonds that hold the protein molecule together called peptide bonds. So you can think of pepsin as the enzyme that breaks peptide bonds. When these bonds are broken, you get chains of amino acids linked together called polypeptides. Now, since we know the prefix poly means many, we can easily recall that a polypeptide is many amino acid units joined together. These polypeptides then move into your small intestine, where digestion will be completed by additional enzymes. Now, in the small intestine, pancreatic enzymes that we previously learned about, called trypsin, chymotrypsin, and carboxypeptidase, really go to work breaking down the polypeptides. And these enzymes enter the duodenum via the pancreatic duct. These pancreatic enzymes are helped by the brush border enzymes. We previously learned that the brush border enzymes are special enzymes found on the microvilli of the small intestine that complete digestion. The peptide bonds holding the polypeptides together continue to be hydrolyzed or broken down and result in smaller units called peptides. Peptides are simply defined as two or more amino acids linked together. Enzymes continue to break down polypeptides and peptides into amino acids. Because amino acids are very small, they're able to be absorbed through the small intestine lining and into your bloodstream. Now, it's important to note that digested nutrients that leave the digestive tract take a detour to the liver before entering the general bloodstream. Your liver is an important organ, and this detour allows your liver to have first claim at nutrients coming from the digestive tract. It's almost like the liver is king, and it gets first dibs at the good nutrients. Then when it takes its fill, the rest of the body has the leftovers. So we see that capillaries in the wall of the digestive tract pick up the amino acids. These amino acids, along with other digested nutrients, then move to the liver through a unique system of veins called the hepatic portal system. This is the system of veins that are responsible for directing blood from the digestive tract to the liver. And we see that the word hepatic means liver, and the word portal means gateway. So you can think of the hepatic portal system as the gateway to the liver. This nutrient-rich blood that drains out of your digestive tract travels to the liver via the hepatic portal vein. This vein is defined as a vein that transports blood from the digestive tract to the liver. Now this is a unique vein. In fact, some would argue that it's not a true vein because it conducts blood to the capillary beds of the liver. We know that true veins carry blood back to the heart, and arteries are typically the blood vessels that travel toward the capillary beds. So the hepatic portal system has many unique circulatory facts to consider. After the liver takes its share of the nutrients, the blood enters the general circulation through the hepatic veins, which drain the liver. The remaining amino acids can now circulate to your body cells. Your cells remove amino acids from the blood and use them to build the specialized proteins that we talked about at the beginning of this lesson, such as enzymes, antibodies, hormones, muscle proteins, or collagen. Let's review. Proteins are defined as a large molecule composed of one or more chains of amino acids. So we see that amino acids are the basic building blocks of protein. Amino acids can be used by your body to form important cellular structures, such as enzymes, antibodies, hormones, muscle proteins, and collagen. Protein digestion begins with the action of an enzyme called pepsin. Pepsin is the active protein digesting enzyme of the stomach. Pepsin acts on protein molecules by breaking the peptide bonds that hold the molecules together. Digestion of protein is completed in the small intestine by pancreatic enzymes trypsin, chymotrypsin, and carboxypeptidase. These enzymes also get some help from the brush border enzymes found in the small intestine. When proteins are broken down, they create polypeptides, which are chains of amino acids linked together, and peptides, which are two or more amino acids linked together. Before entering the general circulation, digested nutrients that leave the digestive tract take a detour to the liver. The hepatic portal system is a system of veins that are responsible for directing blood from the digestive tract to the liver. The hepatic portal vein is the vein that transports blood from the digestive tract to the liver. After the liver takes its share of the nutrients, the blood enters the general circulation through the hepatic veins that drain the liver.
Okay, so to summarize, digestion of proteins really begins meaningfully in the stomach and continues in the small intestine, including important enzymes from the pancreas, through a series of enzymatic reactions that degrade polypeptide chains into amino acids. These are then released into the bloodstream and delivered to the liver, where they form an amino acid pool, are used to make proteins, or are degraded for energy, and the amino group is excreted. And protein synthesis is sensitive to protein influx, which is regulated by insulin. So I forgot to write it on this, so I would like to say before I end that some of this lecture was adapted with permission from Samer Katubi, MD, PhD. And here are your questions, and I hope everybody has a great week. Bye.